And we are rolling. So, and we have another student swelling our ranks. Good morning. And it tells me I'm presenting, so can everybody see this? Should say Protista 2. Am I doing this thing right? Wendy says yes, Savannah says yes. Okay, right. Let's go ahead and rock on to Electric Avenue and continue talking about protists. And you saw this slide on Monday. People are still working out what the really large-scale way is to classify eukaryotes. And the most recent scheme uses these 5 supergroups. Animals are part of one, a group called the Opisthokonta, and we'll talk about them actually for the entirety of the course except these first couple of days. Archaeplastida is plants, green algae, red algae, and a couple of others, and we've got a perfectly good botanist if you want to learn about them. We won't have much to say about fungi, and we've talked about amoebozoa, and excavata we'll talk about on Friday, and today I want to talk about SAR. Now SAR is Straminopiles, alveolata, and Rhizaria, SAR. Straminopiles, I've said all I'm going to. That includes diatoms and golden algae and brown algae and a few other groups like cabbage clubroot disease and potato blight and water molds and things like that. Those are all in the Straminopiles. Rhizaria, the best known groups would be Radiolaria and Foraminifera because they've got such a big fossil record and they're so abundant in the oceans. And you've got prepared slides of those if you care to look more closely. But a really big one is the alveolata, and it's going to take all morning to talk about those. And we divide SAR into Straminopiles, alveolata, and Rhizaria. We divide alveolata into 3 major subgroups, and there's actually a couple that don't quite fit in any of these 3, but these are the big ones. And alveolata refers to the alveolar membrane system because all 3 groups have these flattened vesicles right underneath the plasma membrane. These flat membrane sacs that are just up underneath the cell's outer membrane actually looks a little bit like quilting. If you think of a quilt as, being several sheets of cloth that are stitched together to make these compartments that are, filled with quilt batting. I guess it's not a perfect analogy, but it will do. So you have this kind of quilted structure in alveolates, and they're also similar in their DNA sequences, even though outwardly they don't look that much alike. And the 3 big groups are the Ciliophora, and that includes the ciliates, the ones you're doing this week's lab on, and Apicomplexa is an entirely parasitic group that includes things like malaria parasites, which kill, last I looked, something like an estimated 450,000 people each year globally, and a lot of others. And finally, the dinoflagellates, which you can find in the ocean or in fresh waters. Very important photosynthetic group. And just to show you this directly, on the left, you've got a dinoflagellate called Carlidinium. In the center, you've got that bean-shaped thing is a meront, I think, one of the stages in the life cycle of Plasmodium falciparum. That's a human malaria. And on the right, you've got Paramecium, one of the species of ciliate that you might well see in your water samples. And up at the top, the nuclei have all been stained blue, and proteins that are found in these alveolar sacs called alveolins have been stained fluorescent green. And you can see that all three have these fluorescent alveolin proteins. So there's some direct evidence for this molecular link among these three taxa. And I'll start with the ciliates. And ciliates are called ciliates because they have cilia, which is very profound. No, really, I've got a PhD. Seriously, you know what cilia are, and there's more to it in ciliates than just having lots of them. The cilia are arranged in rows called kinetes. They're not that easy to see, although I'll see if I can set up a demonstration of them tomorrow before we get on to the main business of lab. And every cilium's got this rather complicated arrangement of microtubules that look a little bit like roots that root the cilia in the cytoplasm and that link each one to the other. Those roots are called kinetids. And when the cilia beat, they beat in a coordinated fashion called a metachronal wave. And you know what a metachronal wave is if you've ever been in a stadium and people did the wave, right? People aren't actually moving when you do the wave. You're staying in your seat. You know, you stay in your seat, and then on cue, you get up and you wave your arms. You go, whoa, and then you sit down. But the metachronal wave is that wave of people standing up and saying, whoa, that wave of movement that passes around the stadium, even though the people that make it up aren't changing their positions at all. If you want another example of metachronal wave, I've seen lots of electrical signs that do it. The light bulbs aren't actually moving, but you'll see these waves of light passing around the sign as light bulbs turn on and shut off in a coordinated way. And metachronal waves for cilia are the same thing. The cilia beat like that. The cilia don't actually move, but that pattern moves down the rows of cilia. There, that's the best I can do with what I've got. There's almost always a defined cell mouth called a cytostome. One particular spot where food particles are taken in, and it's not on the slide, but I'll mention that there's also often a spot on the cell where vacuoles that contain waste are expelled. So there's a cytoproct in addition to the cytostome, a cell mouth and a cell anus. And the details vary a lot, but often there's a complex cytoskeleton around the cytostome that allows it to open and close. It may be called the cytopharynx. And or there's often specialized cilia surrounding the mouth that beat in such a way that they set up currents that draw food particles in. And again, the details of this are often important for identifying and classifying what ciliates you've got. Under the surface, you've got specialized organelles that can shoot things out. They're called extrusomes. One particularly common type will, many ciliates can shoot out these little barbed hair-like things, usually in response to poor conditions. Sorry, my wife is giggling at me because the cat is staring at me. You can't see this, but the cat is just going, hey, I want to be in on this. Sorry. And then I talked about this in lab, but there's a dichotomy between a ciliate's micronucleus, which is a pretty well-behaved diploid eukaryote nucleus that goes through cell division, mitosis and meiosis, just like you learned in 1440, and this huge bag of thousands of short chromosomes, each with a few genes on it, each duplicated many thousands of times, this kind of big bag of free-floating genes called a macronucleus, and I'll show you examples of that in a bit. And then finally, there's sexual reproduction, and it's called conjugation, where two ciliates will pair up and form a little cytoplasmic bridge. The micronuclei will undergo meiosis, and then the two ciliates will swap micronuclei and then fuse them together, and so create a new diploid micronucleus that generates a new macronucleus. Ciliates also reproduce by asexual division, but they can't reproduce asexually indefinitely. Typically, after maybe 100 generations, a lineage of ciliates is going to start getting smaller and weaker and eventually die out if it can't find ciliates of the opposite mating type to conjugate with. So here's one of them. This is Tetrahymena. And some of you may have seen this one before because I think it's in Cell. If you've taken Cell, I think they do a lab where you have these guys eating particles of India ink, and then you count how many vacuoles they form, or something like that. I know at least some sections of Cell do that lab. And Tetrahymena has fairly simple cilia. They cover the entire body, and you can see that they're arranged in rows. That's those kinetes. And it's hard to point out, but up at the top and a bit to the right, you have the cytostome. And if the cilia look a little bit tightly packed right around the cytostome, that's because the cytostome is surrounded by 4 membranelles. Tetrahymena means 4 membranes, and it refers to these membranelles. And the membranelles are cilia that are so close together that they beat as one, that they basically do this instead of being more separated and doing that metachronal wave thing. This is also Tetrahymena. It's been stained with a special stain that uses silver. For reasons I don't fully understand, the basal bodies of cilia take up silver. And all of those dark colored dots that you see forming those rows, each one of those dots is a kinetid. It's a basal body anchored by microtubule bundles, although you can't clearly see the microtubules here. Most of that had to be worked out by the electron microscope. And if you want to study ciliates seriously, you have to learn silver staining because the arrangement of these kinetids is used in ciliate identification and classification. And if you're wondering, why should I have to learn trivial stuff like this? Tetrahymena, or at least people that worked with Tetrahymena, Tetrahymena's won 2 Nobel Prizes so far. And one was for the discovery of telomeres, right? These capping structures at the tips of chromosomes. The guy who found that won the Nobel Prize in Medicine in 2009. Understanding telomeres is critical to understanding things like aging. Thomas Cech discovered ribozymes. Those are RNAs that catalyze chemical reactions as if they were enzymes. And he won the Nobel Prize in 1989 in chemistry. The cytoskeletal motor protein dynein, this thing that looks like little legs, that was found in Tetrahymena. Cell cycle regulation, early work on lysosomes. Tetrahymena has been used an awful lot in some very basic cell biology research, and they've been doing it for decades. There's other ciliates that have been used. One called euplotes. was actually used to work out the cell cycle. Some features of euplotes that we don't need to get into actually make it possible to track the progression of the S phase. If you remember that interphase, you have G1, S, and G2. S is the time when DNA is actually replicated. It's possible to see that because of the way that the euplotes nucleus is shaped. So yeah, details of that cell cycle, especially interphase, were first worked out in the 50s in a different ciliate, euplotes. That's why I don't know how many of you want to be doctors or researchers in a different field, but that's one of the reasons why knowing your invertebrates is important, because sometimes they provide the best model systems to study some extremely basic and extremely important biological processes indeed. So yeah, proper respects to Tetrahymena here. Anyway, there you've got a diagram. Don't worry about memorizing all of these words. I'm not going to test you on parasomal sacs and kinetodesmal fibrils and all that. This is mostly there just to show you how the bases of the cilia and the basal bodies are connected to these ribbons of microtubules and microfibers that hold everything in place. You can also see the alveoli. The word alveolus appears up at the top right, and they're very flat, and they're right underneath the external membrane, which here is called the plasmalemma. So that shows you the relations of all these structures. And this happens to be the ciliate euplotes. This one's been stained with a fluorescent stain for alveolar proteins. And if you look at, that's the anterior end, especially if you look up at the front by that zigzag border, you can see this thing that almost looks like a mosaic or maybe kind of irregular quilting. Each one of those little, polygons is an alveolar sac, and you can see how they fit together very tightly in a single continuous layer. This is a really nice picture of what those alveoli look like. Tetrahymena had cilia of all the same length, but there are some ciliates that have cilia of more than one length. This is stentor taken under a scanning electron microscope, and the body's covered with fairly simple cilia. If you look, you can see that they're in those rows, the kinetes. And then there's this kind of horseshoe-shaped, or actually it spirals a little bit, row of much longer cilia that goes around the blunt anterior end, and that sets up these feeding currents that direct food into the cytostome, which is a dark spot down there on the lower right. So there you have these specialized cilia directing food into the cytostome, and that's said, such a ciliate is said to be heterotrichous, meaning different hairs, because it's got two different lengths of cilia. And then hypotrich ciliates can fuse multiple cilia into larger structures. This thing, again, is euplotes, which is probably my single favorite ciliate. And this one's also taken with a scanning electron microscope, and it's got relatively few bristle-like things sticking out, but each one of those bristles is made up of multiple cilia stuck together. These bristle-like structures are called cirri, and euplotes and some other ciliates that you might see, like Oxytricha, Stylonychia, things like that, the cirri are actually rigid enough that the ciliate can walk. Euplotes can kind of scurry around, swinging those cirri back and forth. And then also around the mouth, there's this big zone of a whole series of membranelles. which again beat to create food currents that lead food particles down to the cytostome. So this is just a nice example of what a ciliate can build by sticking multiple cilia together into larger units. If you look carefully at those cirri, you can see that they're looking a little split ends at the end. You're just seeing the fact that they're made of multiple cilia. And then the cytostome, again, that's where phagocytosis takes place. That's the point that's specialized for taking in food. Details are different in different species, but the cytopharynx is a set of microtubules that can open and close the cytostome. These are ciliates that can literally open and close their mouths. On the left, you've got Didinium, which is basically Pac-Man. It's a predator of the ciliate Paramecium. And on the left, that's a Didinium with its mouth shut. The cytostome is at the end of that nose cone up at the top. And here it's closed. On the right, that's a Didinium with the cytostome open. And Didinium can swallow and phagocytose Paramecium, which is at least as big as itself. That's why I said Pac-Man. That cytopharynx can stretch and open very, very wide indeed. I mentioned that ciliates can shoot things out from underneath the plasma membrane. The last time I tried this, I couldn't get it to work, but I've been told if you add a little bit, tiny drop of vinegar to a ciliate culture, it'll kill them, but it will often stimulate them to shoot out their trichocysts. These are just bodies underneath the plasma membrane that Paramecium can shoot out as a defense mechanism. And there you see a Paramecium that has already shot all of them out. Those long, vaguely spiky looking hairs are the trichocysts, not the actual cilia, which are much shorter. A little bit about ecology. There's a few ciliates that are parasitic. This is one you might see if you've ever kept freshwater fish or worked in a pet store. This poor neon tetra right here has a bad case of ick. Ick is short for Ichthyopthyrus, and it's a ciliate that lives for a time on the bottom of an aquarium and then emerges and goes swimming and forms cysts on the skin of a host fish and divides within those cysts and sends out more swarming cells to attack more fish. It can be especially problematic in aquaculture and in the aquarium industries, because whenever you have fish packed tightly together, well, when you have anybody packed tightly together, whether it's fish in a tank or toddlers in a daycare, disease can spread much more rapidly than it could in the wild. And it will kill the fish. When I was a kid, I kept a freshwater aquarium and I lost some fish to ick. There's at least one that parasitizes humans. This is Balantidium coli. It can cause gut ulcers, although mild infections may not have any symptoms at all. And it's transmitted by, again, this is a yucky word, by the fecal oral route. The fecal oral route is, when you get in your mouth something that's been contaminated with feces. So food prepared by somebody who did not wash his or her hands, or getting your drinking water from a source that's got sewage entering in it. Those are common ways that many parasites, not just Balantidium, can be transmitted by the fecal oral route. But yeah, I don't think this is screamingly common, but if any of you become doctors, you might see this someday. There's other ciliates that are commensals, cockroaches. I don't know if I'll be able to demonstrate this tomorrow, but cockroaches often have anaerobic commensal ciliates in the genus Nyctothyrus. And the stomach chambers of cows also contain lots of ciliates. This is one of them, isotricha, from the fluid in the rumen of a cow. The rumen is that first stomach. You know, they've got 4. And up to 50% of the biomass in the rumen can be ciliates. And they play a role in helping break down that food. If anybody has access to a country vet, see if you can't find a way to have him get you some rumen fluid or something like that. I'm told it's pretty amazing to look at under the microscope. Never had the chance myself, I'm afraid. Last time I got really close to a cow was trying to tip it over in high school, and that actually doesn't work really well. Anyway, you get free-living ciliates in fresh waters of all kinds. There's some that can tolerate very anoxic, polluted water, others that like it a lot fresher. They're also common in the marine realm, and this is one of the most common. That's a marine ciliate with a hard covering shaped like a cone. If you look in the wide end of the cone, the cell has retracted a little bit, but you can see the cilia sticking up from one end. This is called a tintinid. And tintinids you need to know because they're critical in ocean food webs. They may exist in up to several thousand per liter. If you take marine biology in the fall, I'm actually going to order some live plankton, and we'll probably find live tintinids. These tintinids graze on small phytoplankton, photosynthetic bacteria that may be 1 micron or less. And you can see those green spots inside the tintinid here. That's food vacuoles. That is very small bacteria and algae that the tintinids are feeding on. And then tintinids get eaten by small crustaceans and larval fish, and those get eaten by bigger fish, and those get eaten by bigger fish, and those get eaten by tuna, and those get eaten by, you know, maybe you for lunch if you're going to make a tuna salad sandwich or something. So very important link in food webs between the primary producers, which are predominantly these really small photosynthetic algae and bacteria, and the higher trophic levels with larger organisms. All right, a little bit about sex before we take a break. As I mentioned, ciliates show nuclear dimorphism, where the micronucleus is your standard eukaryote nucleus, and the macronucleus is this giant sack of DNA fragments. Ciliates can reproduce asexually, but as I mentioned, they can't keep doing that forever. Said 150, maybe 200 asexual generations, typical ciliates will start losing vigor. And the reason is, if you took evolution from me, it's this thing called Mueller's ratchet. What that means is the fact that any asexual lineage accumulates mutational damage to its DNA, and it can't repair all of it. Organisms have DNA repair mechanisms, but they're not 100% perfect. And any organism that just keeps constantly cloning and cloning and cloning and cloning itself eventually accumulates so much DNA damage that it starts losing fitness, and, you know, given enough time, it'll die out. It's a little bit like if you took a picture and you made a photocopy, and you photocopied the copy, and then you copied the copy of the copy, right? Every time you photocopy something, you add errors, little distortions and flecks and things like that. And if you just keep copying the copies of the copies of the copies, you add more at every step. And after a few generations of copying, it's completely unreadable. Same kind of thing can happen with DNA if you never have any sexual recombination. You've got to fix DNA damage somehow, and having sex is a good way to fix DNA damage. Let me put that a slightly better way. The point of sexual reproduction is genetic engineering. You're mixing your DNA with somebody else's, and when you do that, you can produce offspring that inherited better parts of, that didn't inherit your bad genes, but instead inherited your partner's better genes. It's actually a way of DNA damage repair. In fact, we think that's why sex evolved in the first place. And when they do it, it's called conjugation. Two ciliates pair up. They not only have to be of the same species, but they have to be of complementary mating types. And there can be more than two. I think it's Paramecium aurelia has 28 mating types. Each mating type normally pairs up with only one other, but occasionally one mating type will pair up with the wrong one that's not normally complementary to it. So yeah, the sex gets weird when you've got 28 genders. The macronuclei break down, they disappear. The micronuclei divide by meiosis to produce these haploid gamete nuclei. The ciliates swap gamete nuclei. The gamete nuclei fuse into a diploid zygote nucleus. That divides and rebuilds the macronucleus, and then both ciliates separate and divide. So there's a stained ciliate that might be Blepharisma. It's similar to the stentor that I showed you at the lab yesterday. And in this species, the macronucleus looks like a string of beads. And depending on the species of ciliate, the macronucleus could be a sphere, an oval, a chain of beads, shaped like a bar, shaped like a horseshoe, shaped like a backwards number 3, all depending on what species you've got. And it can be an important character for classification. And then there you have two large ciliates that have been caught in the act. There's a couple of big pink, really they are pink, ciliates in the genus Blepharisma caught in the act of going at it. This is, I probably should not be showing you this this picture. This is ciliate porn here. And there you've got a diagram. If you can see that, that pretty much just covers what I've already talked about. But I mentioned that when those micronuclei, when those gamete nuclei fuse, they divide, and they'll become a new micronucleus. And then 4 of those micronuclei will turn into macronuclei, a new macronucleus. And what they do is destroy most of their DNA and duplicate the rest many thousands of times. So in some species, up to 96% of the DNA is maintained in the micronucleus, but it's destroyed when the cell makes a new macronucleus, which is really kind of weird. And this little guy, Oxytrichophalax, is actually being looked at very intently in cell biology right now, because in the micronucleus, the genes are actually scrambled. They're not continuous DNA sequences, one from the other, and one after the other. And when the micronucleus divides to make the macronucleus, the genes have to be spliced together to make a functioning macronucleus. It's really weird. that these guys do this very complicated splicing trick to make the macronucleus, and there's quite a bit of interest as to how and why they do it, and what this could tell us about DNA editing and splicing. So, if you're interested in cell biology, there's a number of really cool papers that have come out about Oxytricha phallax. Okay, right. Let me see. I'll check that. Okay. Any quick questions on the chat? Okay, I'll go ahead and stop recording for now, and we'll stop for now.